Hello everyone, I'm Mr. Furlong, and today we're talking facilitated diffusion. We've been focusing on this analogy that cities are like cells. Cities need to get things in and out of it, just like cells need to get things in and out of it. So we're going to be focusing specifically on things traveling through proteins in the cell membrane. Now remember, there's many different types of proteins that can be found embedded in the cell membrane. One type are transporter proteins. So this would be things that molecules can move in and out of the cell through. But there are others, like there's receptors on the cell membrane. We'll talk about those later on this year. Some act as enzymes. Others act as anchors so that they can connect one cell to another cell. So there's lots of different types of proteins. We're going to be focusing on the transporter ones in this video. Now remember that facilitated diffusion is the movement of molecules from areas of high concentration to areas of low concentration, but it needs to go through a protein. So these are big molecules, too big to go through the cell membrane, and depending on the molecule, it goes through a specific type of protein. Because molecules are moving from areas of high concentration to low, this is not going to require any energy. Osmosis, the movement of water, is one type of facilitated diffusion. Remember that the tails of lipids are hydrophobic, so they don't want to be in contact with water. So water travels through a special type of protein called an aquaporin. And there's millions of these aquaporins on the cell membrane that can easily allow for water to pass through. Now, larger polar molecules also need to go through a carrier protein. And these can be opened or closed as needed. So if we need certain molecules, they'll open up and molecules can either enter or leave. If we don't need them, the proteins can close up. Since there is an electrical charge on the cell membrane, this can also allow for various charged ions to move through the membrane as well. Now, large nonpolar molecules can also get in via carrier proteins. So an example of this is what's called the glut protein. And this is what allows glucose into the cells. Of course, glucose is going to be used in the process of cell respiration to provide us energy. These carrier proteins are specific to the molecule. So this glut protein will only allow glucose in. And this is how the cell can be selectively permeable. Nothing else can pass through that protein except for glucose. Now we know that membranes can become polarized just by the movement of ions across the membrane. So in this example, we see a part of a neuron, a nerve cell. And it is positively charged on the outside, negatively charged on the inside due to the number of ions that are there. When a nerve impulse comes, some of these ion channels open up, and for a split second, the outside becomes negatively charged and the inside becomes positively charged. And then other ions open up, and it quickly reverses back to a large positive charge outside and a large negative charge inside. Over time, it eventually repolarizes itself back to its normal positive outside and negative inside until the next nerve impulse comes along and changes it again. Now, some membrane proteins are needed for active transport. Remember, active transport is the movement of molecules from low to high concentrations. This requires energy, so it's getting its energy from ATP. And a good example of this is the sodium-potassium pump. Oftentimes, we need to pump sodium from low to high and potassium from low to high. And so one protein can do both of these. So here we see sodium moving into special areas in that protein. Along comes an ATP molecule, drops off a phosphate, which will change the shape of that protein, which will then release those sodium ions, in this case outside the cell, so it's going from low to high. Now while it's open, some potassium ions move into that channel. The phosphate is removed, which changes the shape of the protein, and now the potassium ions can be moved inside the cell, again going from low to high. Also notice that for every 3 sodium ions that move through, only 2 potassium ions move through. Now this does require ATP for that energy, but it also needs an enzyme in order to break that phosphate off of the ATP. The enzyme is called the sodium-potassium ATPase. And this is the enzyme that breaks the phosphate off the ATP and can maintain that membrane potential. So here we see that ATP molecule. Now, not shown on here would be an enzyme that is going to break off that third phosphate so that it can attach to the protein, forming ADP. Now, this helps to maintain the membrane's potential in neurons, which goes back to that video that we just saw. Now, mutations in this gene can cause neurological diseases. So we can see the importance of sodium potassium ATPase. If there's not enough, you're not going to be moving much sodium and potassium, which could be potentially dangerous. So that's a little bit about facilitated diffusion, and I'll see you in class.